Hello students, Namaste. Today we are going to continue the same unit of grade 8, that is living beings and their structures. Before it, in this unit we have studied the introduction of cell, its different shapes and sizes, and some fundamental components of it. Yes, and fundamental parts you can say, in which we have discussed about the outer covering, that is cell wall, cell membrane, and then we discussed about cytoplasm, and we have studied about their structure also. Yes, today we are going to continue it, and under it we will discuss about the remaining cell components. Okay, so let's go about a question that is very interesting question. At first you see the diagram. What is it? Is it straight? No, it is bitter, yes. And this green, here are shown green chilies. And you know that green chilies turn into red? Daily we see that about tomatoes in different types of fruits. Yes, in many things we found that when they are raw, they are in one color, and when they ripen, they change their color. So question is related with it. It is, how do green chilies change into red when they ripen? Interesting. So in today's lesson we will discuss, yes, what component of a cell, what cell organelle is responsible for it. I think you can guess also a little bit because of that green color, red color. In class 7 also you have done something about cells. So some guess you can do. It is related with plastids. Yes, in fact plastids are of different types, and out of them green colored plastids are chloroplast, and chloroplasts may change into chromoplast also after certain chemical processes in them, and they form chromoplast. So that happens in raw fruits, in as well in chilies also, when they ripen and they change into another one and the color is changed. Okay, so let's see, we will discuss about plastid and its different types later. At first a simple activity is given, because last time you did not practice about the cell's structure, only fundamental things we have studied. Today we are going to discuss about, we are going to write and we will learn the simple method of sketching of animal and plant cell. But at first I will tell you about a common cell, that is same cell in which both parts are shown, but really they are not found, and later you will make animal cell and plant cell separately. Okay, consider. So now see here the shape I am giving of irregular shape, that is an animal cell. In fact it is generalized cell. Okay, and here nucleus you can make very easily, the round structure, inside it some thread like structures, yes, then some fluid like structure. What are they? Later we will study, don't worry. Here it is complete nucleus, yes, and nearby it you have to make, what is it? Can you guess what is structure? Yes, they are what? Endoplasmic reticulum. You can make in this way. Endoplasmic reticulum may be at other places also, like that. Not necessary that only here they are found. They may have some rounded structure on them. What are they? Those structures who are complex to sketch, I will give you more time. But here these structures, these lines and dots, you can sketch very easily. So do with me. I am doing here, you also see and do after me. Yes. So two structures of cytoplasm we have done. Cytoplasm I will fill later. Endoplasmic reticulum and ribosome. And here endoplasmic reticulum is of other type, that is smooth endoplasmic reticulum. You can make vacuole. Vacuole is also easy to make. Now mitochondria. For mitochondria, you have to make elongated structure, yes, and that is outermost layer. Inside it, next layer in this way. Look, equally to that layer and then go inward. Again, is it clear? You can try. Yes. Look here. Yes. In between these two projections again. Yes. In fact, this structure is visible under electron microscope only. Yes. Here, if we are showing them clearly, that shows the diagram nice. So you can make it at different places. Different places. 2, 3, enough. Yes. I'm not going to use that color. With black also you can, because you are also using pencil. Yes. You can make it in this way. Yes. That is mitochondria. Mitochondria. Yes. So you can sketch it. Only one was enough to practice. After that, you can sketch nicely about mitochondria. Yes. Mitochondria. Yes. Now one Golgi body you have to show. How to sketch Golgi body? Make one biconcave plate-like structure like that, and some more biconcave plate-like structure in this shape. Here, biconcave means thinner at middle. Yes, that is what? Golgi body. Then you can make ribosome at other places also. Ribosome. Okay. As I told you, plastid and plastid is not found in animal cell, but for easy, just to practice, I am making here plastid also, similar in shape with mitochondria. Yes, similar to that. And here you can make this structure. Look, that is what? Plastid. So here I am saying, as I told you, that it is just imaginary cell. Just we are practicing different parts. And in the case of Euglena, that is found also in its cell, chloroplast. So in this way, you can show 2, 3, 4, or only 1, 2 enough. Okay. Next is just round with some dot-like structures in it. What is it? Yes, it is lysosome. Lysosome. Another structure you can make in this way, not necessary to make as it is printed in book, very simply, like owl's eyes. That is what? Centrosome. Yes. So major structures are finished, and now fill the remaining space with cytoplasm. Yes. In this way, how nicely you can sketch the diagram of a cell. If you are sketching animal cell, same shape you have to give. If you are sketching plant cell, then little bit. You can say that rectangular or hexagonal shape you can give. Yes. And some particular structures you will not show there. What are found in animal cell, what are found in plant cell, you know, and you will learn also. Don't worry. And then labeling is the main thing, students. Sometimes students make the labeling very poor and the diagram does not look nice. So arrows should not be crossed. Yes, arrows should not be crossed. And the line of the labeling should be the same. That is cell membrane. It is vacuole, mitochondrion. Singular is mitochondrion, students. Plural mitochondria. Lysosome. Nucleus. In fact, different parts of nucleus you will identify later. Only as a whole I am writing a nucleus. Then Golgi body. Plastid is here. Some more structures are here. You can write it endoplasmic reticulum, endoplasmic reticulum, and then you can label it as ribosomes. Okay. In fact, I have sketched a generalized cell. Generalized means usually they are not found. I am showing both the structures found in cell. Here you can show cell wall also above it, doesn't matter. If you are writing a thick layer around it, that will be cell wall. Yes. So it is generalized cell. When you will sketch animal cell, then you will not show centrosome, you will not show cell wall. If you are sketching plant cell, you will give particular shape as I told you, and then you will show plastid there. You will not show there what? Centrosome. Yes? Okay. So that thing you will keep in mind. And later, here I will show you, here look here, this diagram. Yes. Same structures are given here. Yes. So now I think you can sketch easily as animal cell and plant cell. Yes. Different parts are given here. And I think this practice will support you a lot. Yeah? Okay. So you can see what structures are common in plant cell and animal cell. Here in plant cell, chloroplast is found, or you can say plastid is found, but in animal cell, plastid is not found. Here centrosome is not shown, but centrosome is found in animal cell, not in plant cell. Cell wall is found in plant cell, not in animal cell. Other things are common. Both have Golgi bodies, both have lysosomes, both have endoplasmic reticulum and ribosome, both have vacuole, both have nucleus. Yes. So mitochondria, both sides. Yes. Okay. Now we are going to see in detail about mitochondria. Mitochondria structure. Look here, that structure we have practiced. Different parts are given, which is not necessary for your level, but very simple part I am introducing here. That is inner membrane, and this one is outer membrane. Easily you can identify. And the zigzag structure inside the mitochondria is called as cristae. Yes, and here are found ribosome, DNA. What is DNA? DNA is deoxyribose nucleic acid, a chemical. Yes, that name will be given. I will give you the full name later. I think under it you will get the name. So that is the structure of mitochondria. Yes, mitochondria are long, rod-shaped and or cylindrical in shape. Rod or cylindrical shape you can say. Yes. Okay. Many mitochondria are found in a cell. And another thing you are clear already, you have found that it consists of two layers, outer membrane and inner membrane, two layered structure it is. Yes, double layered membrane. Yeah, and it conducts cellular respiration in it. Inside it, cellular respiration. Respiration is the process by which food is changed into energy. The process of changing food into energy is called as what? Respiration. So because it occurs inside the cell, that's why we can say that it is cellular respiration and liberates energy for the body. Due to this reason, it is considered as what? Powerhouse of a cell. If you are asked that which cell organelle is powerhouse of a cell, then that is what? Mitochondrion. Mitochondria are powerhouses of the cell. Okay. Now, what is its function? Some function you have found. Again, mitochondria form ATP. ATP means adenosine triphosphate. ATP is adenosine triphosphate. Yes. So when food is digested and food is reacted with oxygen, after the process of cellular respiration, a chemical is formed, that is ATP. And this chemical changes into what? ADP. Here is adenosine triphosphate. This one is adenosine diphosphate. And during that, energy is released. And this energy is used for different life processes. This energy is used different mechanical work. Yes, we are standing, we are walking, we are talking. That is because of this energy. And that energy is changed form of ATP into what? ADP. And this ADP again uses the energy from food and it changes into what? ATP again. That is stored in our body. So how easily the cycle runs in our body? ATP into ADP, ADP into ATP. ATP changes into ADP to liberate energy for different tasks, and ADP uses something from food and that forms ATP again and stores in our body. So that storage is inside mitochondria. Production and storage both is inside mitochondria. That's why mitochondria is called as what? Powerhouse. Mitochondria is not singular, mitochondria are called as what? Powerhouse. Okay. They also contain DNA. I told you that DNA, I will give you the name, deoxyribose nucleic acid. Deoxyribose nucleic acid, DNA. Another is RNA, ribose nucleic acid. Here we are talking about DNA, which transmits parental characters into offsprings. So DNA is carrier of parental characters into offsprings. Mainly it is found inside nucleus and the chromosomes, but its location is found at other places also. In plastid also it is found, its little amount. Its little amount is available in mitochondria also. So there, from there also they can transmit what? Parental, they can carry parental characters into offsprings. Okay. DNA are carrier of parental characters into offsprings. Clear? After that, we are going to discuss about plastid. Plastid, how simple diagram I shown you. Here it is very complex. Yes, you can use simple diagram as I taught you. So it is also double layered, students. Outer layer, and that is outer membrane, and it is inner membrane. Two layered. And in it, you are seeing the pile of coins. In fact, they are not piles of coins. These coin-like, each coin-like structure is called as thylakoid. Thylakoid. And the pile of thylakoid, which looks like the pine of coin, that is called as what? Grana. Singular granum. Granum, and all of them are grana. G-R-A-N-A. Grana. And they are connected, from each pile is connected with other pile by lamella, and the space around the grana is filled with stroma. Not difficult, not necessary to memorize each part, but if you like to be master, you have to learn something, you have to know something more. Yes, so it is not difficult to understand these things. Yes, outer membrane, inner membrane. Yes, inside it the pile like structure is of thylakoid. Each thylakoid, each coin-like structure, and the whole pile is called as what? Granum, and the group of granum is called as grana, and the remaining space is filled with stroma, in which DNA may be found, in which chloroplast may be found, or you can say chlorophyll may be found. Yes, so different things are also found. Don't go in detail. That is enough for you. Plastid is made of double layered membrane. It is found in plants, few bacteria, and euglena-like animal also. In animal also plastid is found, but they are just exceptional case. Euglena, a protozoan, you have studied about. It is concerned with preparation and storage of food. It prepares food, that is chloroplast, and it is stored also in the cell by leucoplast. Chromoplast also support in different ways. There are three types of plastids which are given here. You can see that it is of three types: chloroplast, chromoplast, and leucoplast. Chloroplast having green pigment, chromoplast having other than green pigment, and leucoplast, here it is shown purple, but in fact it does not have any color. while seeing under microscope, it is stained by some chemicals to see it clearly only. Okay, it is colorless. It has no color. Okay, in detail we will discuss. We are going to study in slightly detail about these three types of plastid. First one is chloroplast. Yes, chloroplast, and its function, it is green. Green plastid has chlorophyll in it. Yes, that is green pigment which is found in green parts of plant, usually in leaves, which may be in other parts also. Some fruits also, when are exposed in sun for long time, they become greenish. Potato, that is root. Yes, that changes into green also. You have seen some green potatoes. Yes, so they may change. One type of plastid can change into other, but not all. Here, this chloroplast can be changed into chromoplast. Already you have seen in the case of chilies. Yes, yes. Leucoplast can change into chloroplast also. Leucoplast, that is non-green potato, may change into green. Sometimes it happens. In few cases it happens. Okay, chloroplast has chlorophyll, which is green pigment. Chlorophyll is green pigment. It is found in different shapes, usually oval, but ribbon shaped you can say in Spirogyra. Spirogyra is what? That is a type of algae. And another organism, do you know? Chlamydomonas. Yes, in Chlamydomonas it is cup shaped. Cup shaped. Yes. So in different shapes chloroplasts are found. Yeah. It contains double layered membrane around it. Yes. And chloroplast is responsible for photosynthesis and makes plant green. Due to the presence of chloroplast or chlorophyll in the chloroplast is green. And due to the presence of chloroplast, plant is green, leaf is green. Different parts of plant where they are found, they are green. Yes, in some cases stems are also green. In some cases fruits are green. Many cases when they are raw, they are green. Yeah, okay. So it is green pigment, and that green pigment supports in photosynthesis because it utilizes the solar energy. It absorbs the solar energy to change one type of chemical into other. Yes, by mixing or by combining water and carbon dioxide, glucose is made in photosynthesis. For it, the solar energy is used, and it is the chlorophyll, chloroplast, which makes to use the solar energy for this purpose. Yes, about it you are doing from very junior classes. So just you recall this knowledge. It forms fatty acid and amino acid in plants. Fatty acid is a basic structure of fat, and amino acid is basic structure of protein. So it forms fatty acid and amino acid from which protein and fat are formed. So fundamental formation of photosynthesis is glucose. From glucose, different other food materials are formed. It also contains DNA that transfers parental characters into offsprings. Already I discussed that DNA may be found in other part than nucleus too. Yes. Okay. So next plastid is what? Chromoplast. Chromoplast is another type of plastid which is colored but other than green. It is not green in color. It may be red, maybe that is orange, yes, maybe yellow. In different colors it is found, and it provides attractive colors to fruits. Flowers are very attractive. Yes, and this attraction is given by colors also. Yes, we love flowers. We like flowers. Yes, so that attractive colors of flowers is because of the what? Chromoplast. And not only that, as we discussed about the chilies, tomatoes, they also have what? Chromoplast. Chromoplast is found in carrot also. In beetroot also, there are what? Chromoplast. Yes, and remember that chloroplast also may change into what? Chromoplast. Yes. Okay. Chromoplast is a colored plastid other than green. Mainly it is found in flowers, ripened fruit and other colored part of a plant. Okay, what is the advantage of providing attractive colors of flowers? Attractive colors of fruits? Disadvantages to plant. A plant has attractive flowers which is supported for what? Pollination. The organisms are attracted by that flower and they do pollination in them. So that is one way of advantage of color in plants. Fruits, that attractive colors, by attractive colors they can be identified easily, their location, and they are picked from there. You know that animals pick them and they take it away from the plant, eat the mesophyll, the main part, edible part, and leave the hard seed there. Yes, so that is about dispersal of seed you have done. So food is dislocated from where it is, that means born. From there it is dislocated to another place and seeds are transmitted from one place to another place. Yes, so that is supportive in dispersal of seed also. It is slightly flat and has colored pigment. Yes, we know it, color pigment, but its shape, here you have to note it in your mind that it is not as swollen as what? Chloroplast. It is slightly flat. Yes, already we have finished. This one also we have finished. Yes. Okay. It is also found in the root of some plants like carrot, beetroot, etc. That helps in their growth. Yes. Here the chromoplast found in the root of beetroot and in carrot helps in growth of this root. Yes, you know that their root is the growth of these roots is abnormal from others. Yes, so that is supported by it. It's not only that, but it is just supporter. It supports in the growth of root of carrot, beetroot, etc. Okay, leucoplast. That is the third one, plastid, and this plastid is not colored, colorless, found in roots. We talked about special type of roots. Here in normal roots it is found. Leucoplast is colorless plastid and does not contain any pigment. Pigment means coloring thing, coloring object, coloring agent, pigment. So pigment is not found in it. It is about round in shape. Yes, round, oval. It is found in root and other colorless parts of plants. Yes, not in flower and fruits. Its major function is to store different types of food like starch, protein, fat, etc. in the cell. If you study in other sources of that means Google, etc., then you will find that leucoplast is further divided into three types. One type is that which stores carbohydrate. Another type is that which stores protein. Another type is that which stores fat. So for the storage of these different types of food, different type of leucoplasts are found. So they are categorized on the basis of their function. Okay. So anyway, they are concerned with what? Storage of different type of food. Okay. So in this way we have finished about two cell components till now, mitochondria and plastid with their types. Now next one. Oh, it is very different diagram from your book, students. I think it is simple for you to sketch. Yes, the diagram given in your book, that is also okay, nice, but there are some terms which are not understood by you. Yes, that is what is mean by 70 years, what is mean by 80 years, that is also given above the diagram. You have seen in your book, yeah. In fact, this is a thing of higher level. 70S means that S is sedimentation capacity, and it is short form of Svedberg. 70 is Svedberg. That is not necessary for your level. Up to STE also that is not required. Yes, but because it is given in your book, you should know that what is 70 years, what is 80 years, why it is given there. So, for your exam, not necessary. For your knowledge, you can say that it is 70 Svedberg and it is 80 Svedberg, and it shows the sedimentation capacity of that particle. And one special thing about ribosome is that only that is, till now we have studied, what? Without membrane, no single membrane or double membrane, coverless. Coverless structure is that. Now, say about ribosome. Ribosomes are usually found freely in cytoplasm, but you have seen that it is found attached with what? Endoplasmic reticulum. It is found inside other structures also. So, mitochondria, yes. So, like that it may be found at other place also, but mainly they are concentrated in what? Cytoplasm, and sometimes on what? Endoplasmic reticulum. Yes, so mitochondria, plastid, yes, already you have finished. They do not have any covering membrane. I told you, yes. So, without covering, ribosome, remember it, unique. They form protein by the process of protein synthesis. That's why they are called as protein factory of cell. Protein factory of the cell. Yes, okay. They also participate in gene expression. Gene expression means genes are what? I told you DNA. DNA is the main component of gene. What is the function of DNA? Transformation of what? Parental characters. So, these genes carry some characters which are expressed into, which express their characteristics in offspring. So, that gene expression is controlled by what? Ribosome. Yes? Okay. So, next one is endoplasmic reticulum, and in endoplasmic reticulum is found around the nucleus, and that connects nuclear membrane with what? Cell membrane. It provides internal support also. That's why sometimes it is considered as skeleton of a cell. Endoskeleton, like endoskeleton, we have skeleton inside our body that is supporting part of our body. In the same way, it supports the cell. Okay. So, endoplasmic reticulum is long, branched, double layered network like a structure is spread in between nucleus and cell membrane. Inner part is hollow. Its inner part is hollow, and outer surface may be connected with ribosome. Maybe, not always. Look here, it is not connected with ribosome, but here connected with ribosome. So, that part which is connected with ribosome, it is called as rough endoplasmic reticulum, rough ER. And that one which is not connected with ribosome, that is called as what? Smooth ER, or smooth endoplasmic reticulum. So, endoplasmic reticulum may be rough or smooth. Okay. Now, what is the function of endoplasmic reticulum? Endoplasmic reticulum makes a structural framework of a cell, already we discussed, that is endoskeleton of cell. It provides internal support. And another thing, helps in metabolism in cell due to the presence of enzyme in it. It also contains enzyme that support in what? Metabolism. Next thing, it helps in internal transportation also, students, because they connect cell membrane with nuclear membrane, cell membrane to nucleus. So, in between them substances also can flow through this region. Yes. So, that supports in what? Internal transportation also, cellular transportation also. But here is another thing also given, supposed to form nuclear membrane and Golgi body. It also forms nuclear membrane and Golgi body and participates in cellular transport system. Yes. So, that is about function of endoplasmic reticulum. So, in today's class we have discussed about what? Mitochondria. Then we discussed about ribosome, endoplasmic reticulum, plastid and their types. Yes. So, I think you have understood well. Let's see how much you have learned. So, before that, here to strengthen your study, you have to do one project work also. you have to sketch a color diagram of animal and plant cells. At same place, you have to sketch the diagram, animal cell and plant cell, and you have to do common labeling in between them. Those structures which are common, give label 1, only arrow 2. And those which are not common, use dotted line. That will be more clear for what? Difference and similarities. Okay. So, after that, let's see how much you have understood. Which cell organelle is considered as powerhouse of a cell? Ribosome, Golgi body, endoplasmic reticulum, or mitochondrion? So, I think easily you have guessed it is what? Mitochondrion or plural mitochondria. Now, next question is, which of following is cell inclusion? Is mitochondria cell inclusion, Golgi body cell inclusion, gas vacuole cell inclusion, or ribosome cell inclusion? About vacuole you have not studied, but we have discussed that these parts are found in cell. Yes, so gas vacuole is what? Cell inclusion. Next question is, which of following statement is true about cell organelles? Mitochondria support to form DNA in the cell. Is it? Ribosomes support to form protein in the cells. Is it true? Think once. Smooth endoplasmic reticulum has ribosomes in it or on it. Chloroplast can change into leucoplast inside cell. So, I think little bit difficult, but you can find the answer. That is what? Ribosomes support to form protein in the cell. Mitochondria does not support to form DNA. Smooth endoplasmic reticulum does not have ribosome. Chloroplast cannot change into leucoplast, but leucoplast can change into what? Sometimes chloroplast. In some cases it is found. Yeah. Okay. Now next question is little bit more typical. So not difficult, but typical. Just you have to think, you have to find out 2 columns. Column A, column B. Yes. What is given here? What is given here? Now matching. Which matching is correct? Find out. Yes. Absolutely, that one is correct. 1C, powerhouse of a cell is mitochondria. 2D, protein factory of cell is what? That is ribosome, D. 3A, may be rough or smooth, endoplasmic reticulum. And 4B, root of plant, that is leucoplast. Okay, students, now there are some questions to which you have to copy and answer them to show your teachers. Write 2 similarities and 2 differences in between plastid and mitochondria. You have to try and you can do it. Yeah. Next, name any 2 organelles made of 2 layers. Some organelles are made of 2 layers, some organelles have single layer, some organelles have no layer. So, 2 layered organelles you have to name. It is simple. What is function of ribosome? Main function you have to discuss here. Which organelle is considered as non-layered organelle? Also write its function. Yes. Answer the questions based on the given diagram. Diagram are of you identified. Yes, chloroplast, chromoplast and leucoplast. First question is, which of them is found in the root of carrot? You can guess. Yes. Which of them stores food in cell? Yes, which is related with the storage of food. And finally, describe in short about A with its 2 major function. A, not only A. Yes. So, about it you have to describe and 2 major functions you have to write there. Okay, students, that's all for today. See you in next class. Namaste.